Hello everyone, welcome back to another session of regeneration biology. So today we will learn about different types of cells, and they are broadly classified into totipotent, pluripotent, multipotent, unipotent, and we will try to understand these nomenclature in a regeneration context and why they are named that way. So what is a stem cell? And we know that a cell that has the ability to continuously divide and differentiate, that is capable of developing into various other kinds of cells, and that is what is called a stem. We know stem, the concept which is able to anchor some group of branches, etc. That is the whole word stem emerged from. Stem cell, it can make a copy of itself, and that is self-renewal, and then it can give rise to another cell type, differentiate. So if a given tissue has got 100 cells, if 30 of them are deciding to differentiate, then the stock should remain back to 100. How is it possible? This 30 will divide and replenish back the 30. So 30 will divide into 60. In that 30 will stay back in the pool. Remaining 30 will differentiate. This is how usually done. So normally the stem cell pool has to be maintained perfectly. So stem cell has the potential for differentiation, and based on the potency of differentiation, we have classified them. Potency is basically a measure of how many types of specialized cells a stem cell can make it. Totipotent can give rise to a complete individual, not just complete list of tissues present in an organism, rather a complete individual. Say for example, you have got heart, liver, kidney, muscle, brain, etc. If I take all these tissues and pile together, put in a bucket, it won't become an organism, right? So forming an organism is slightly different, and we can even call that, that is a congregation of all these tissues and giving rise to an organism. So totipotent cell is capable of doing that. And these cells are from around very early developmental phase. They have this ability, but after a few days, little bit while, they become pluripotent. That means they can make all cell types or specialized cells in the body, but it cannot make the entire organism. That is, embryonic stem cells are normally referred to as pluripotent cell. They are not totipotent. They cannot give rise to an organism. Multipotent can make multiple types of specialized cells, but not all types of cells present in an organism is not possible. Classic example is a tissue-specific stem cell, bone marrow stem cell. They can give rise to WBC, RBCs, etc., but they cannot make your skin cell. Unipotent stem cells that produce one type of cell which are basically capable of renewing themselves and also distinguishes from other cell types. For example, muscle stem cell. If there is a tiny damage to the muscle stem cell come into picture and give rise to only muscle, not neuron or some other cell type. So important characteristics of stem cells. One is self-renewal, that is, they are capable of making copies of themselves, renewing themselves, because the stock has to be maintained. And regeneration. They can be induced to become tissue or organ-specific cells with specific functions. In some organs, such as the gut and the bone marrow, stem cells regularly divide to repair and replace worn out or damaged tissue. We have seen our intestinal epithelium is always under erosion, and the intestinal stem cells give rise to the intestinal epithelial cells. In other organs, however, such as the pancreas and heart, stem cells only divide under special conditions, and that too in special organism. Many animals, they will lead to scar formation. Types of stem cell. Embryonic stem cells come from 5 or 6 day old embryo. That is basically the embryonic stem cell. They have the ability to form virtually any cell type of that organism. And embryonic germ cells, they are derived from the part of human embryo or the fetus, will ultimately produce eggs or sperm. They are called embryonic germ cells. And then adult stem cells, they are undifferentiated cells found among specialized or differentiated cells in a tissue or organ after birth. That is what we see it. Like we know liver has got stem cells, also we have got skin stem cell, we have got a muscle stem cell, and even some retina, rods and cones, we have got rod progenitor cells, like that. They appear to have more restricted ability to produce different cell types, and also they are capable of self-renewing. And induced pluripotent cells, they are the cells, they are adult cells of the body which are reprogrammed to show pluripotency. That means you take the skin cell and you express some transcription factors. Now you convert them into stem cell, which is induced pluripotency cell. So you can see in this cartoon that egg and sperm fertilized 2 cell, 4 cell, and this is a group of cell morula stage, and you can see this is a blastula stage, and inner cell mass of the blastula, they are basically embryonic stem cells. You can collect from here and then culture in a Petri plate, and outer layers of cells usually become the trophectoderm. The inner cell mass is usually used as embryonic stem cell, and tissue stem cells are present in the embryo, and adult stem cells are present in the adult for the maintenance of a given tissue. Embryonic stem cells, where we find them, and like I told you, it is the inner cell mass of blastula, and they can be separated and cultured on a plate, and you have to give fluid nutrition, and also you should have some leukemia inhibitory factor, or you should have a feeder layer in the bottom, because which has to provide the growth factors that is needed for maintaining the stem cell as stem cell. And upon some insult or some differentiation cues, if you give, it can give rise to muscle, neuron, skin, etc. Whatever you want, you can make it differentiated into all possible types of specialized cell. So embryonic stem cells, and they are pluripotent cells derived from the inner cell mass and of the blastocysts, and they are derived at a particular stage of the embryonic development. You cannot go to any stage of the embryonic development. And details about the blastocyst and also the embryonic stem cell source. The blastocyst is a structure formed in the early development of mammals. It possesses a part called inner cell mass, or called ICM, which subsequently forms the actual embryo of that particular species. The outer layer of the blastocyst consists of cells collectively called trophoblast, and this layer surrounds inner cell mass, and a fluid-filled cavity is there in the blastula, which is known as the blastocele, and the trophoblast give rise to the placenta eventually for inner or internally developing organism, because uterus and placenta are important for the development of the embryo. In humans, the blastocyst formation begins about 5 days after fertilization, when a fluid-filled cavity opens up in the morula. A ball consisting of a few dozen cells exists in that phase. The blastocyst has a diameter of about 0.1 to 0.2 millimeter, and it contains around 200 to 300 cells. Following rapid cleavage, it forms at a rapid pace compared to a normal skin or any other cell type. After about 1 day, the blastocyst embryos itself into endometrium, embeds itself into the endometrium, that is the lining of the uterine wall. Where it undergoes later developmental process, including gastrulation, and this is supported by the placenta formation at a later stage. The inner cell mass of the blastocyst is a source of embryonic stem cell if you want to create from that particular species. Embryonic stem cells, they have unique characteristics. They have self-renewal in an undifferentiated state for a long period, and the maintenance of stemness. Stemness means what? Ability to give rise to any cell type, or pluripotent markers. They will express like SOX2, OCT4, KLF, CMYC. These are all some of the, they are called, Thomson factors or Yamanaka factors. They are used for making stem cells from differentiated cells. So they are hallmarks of embryonic stem cell. And formation of teratoma, which induced in severe combined immunodeficiency mice, called SCID mice. So what is teratoma? Any cell you implant into a immunocompromised mice in the uterine environment, that cell, it's basically a cancer that will have all tissues that is formed, that has got teeth, nails, skin, bone, etc. But it's not an organism. Why? Because it is not formed from a zygote. It is formed from a malformed cell in the uterine wall. So that is called a teratoma, and this is done in SCID mice mainly because you don't want your immune system to get rid of that unwanted tumor. Normally, a malformed embryo will be taken care of. Taken care of means by expulsion by the immune system of the mother. So if that has to be compromised, then teratoma can develop. Maintenance of normal karyotype. Karyotype means the number of chromosomes. 46 chromosomes we have, 23 pairs. So that is the karyotype of human. That should be maintained as it is. Because of division, we cannot change. And clonality. Clonality means ability to make a exact copy of themselves. So stem cell markers expression, nano, OCT4, don't think these are the two markers, there are many markers, but these OCT4, nano, SOX2, these are all seen as markers of stem cells. So clinical research, it is huge implication in clinical research. Myocardium disease, that is the regeneration of the damaged heart muscle by injecting human embryonic stem cell derived cardiomyocytes directly into the site of infarction, can have some beneficial traits that develops because of the stem cell delivery. Lung disease, alveolar type 2 epithelial cells derived from the human embryonic stem cells, and in a nude mice model. Nude mice, again, you don't have the immunity. That means it do not reject the foreign cell. That is called a nude mice model of acute lung injury method, also has been published. Nervous system, that is oligodendrocyte progenitor cells derived from the human embryonic stem cells, can improve functional locomotor behavior after cell implantation in the damaged site 7 days after the injury, basically in the animal model. So a lot of implications are there for these stem cell research. Human embryonic stem cells are generated by transferring stem cells from the pre-implantation stage embryo. Before pre-implantation means before it is getting attached onto the uterine wall or endometrium, into the plastic laboratory culture medium and dish that contains a sufficient quality of culture medium. The inner surface of the culture dish is coated with mouse embryonic skin cells, specifically treated so that they will not divide. They will just survive. They produce the nutrition or secrete growth factors for the stem cells to thrive. This coating layer cells is called the feeder layer. On top of that, you can grow the stem cell. Cells divide and spread over the surface of the dish, and the plated cells divide and multiply and crowd the dish, and then they are removed gently and plated into several fresh culture dishes, which is also having this feeder layer. This process of replating or subculturing the cells is often referred to as passage, not passage, passage. That is what it is referred. You can keep going. And once the cell lines is established, the original cells yield millions of embryonic stem cells that have proliferated in a culture for a prolonged period of time without differentiating. And the pluripotent cells are often referred to as the embryonic stem cell line. When you can go into different passage, we often refer to them as human embryonic stem cell line. Importance of a feeder layer. The mouse cells in the bottom of the culture dish provide the cells a sticky surface to which they can attach. Also, the feeder layers release nutrients into the culture medium, which is beneficial for the embryonic stem cell. Disadvantages of feeder layer. What is the disadvantage? There is always a risk that viruses or other macromolecules in the mouse cells may be transmitted into the human cells, because even though it is not dividing, it is vulnerable to release some unwanted biohazard into the stem cell. So if you are using for human treatment, one should be wary about that. Differentiation of the embryonic stem cell. So fertilization, embryo, group of cells, inner cell mass. Collect it, culture it, and you keep passaging it, and you can give rise to ectoderm, brain, skin, mesoderm, muscle, blood, endoderm, lung, gut, and germline, sperm, egg. You can make anything. Just your imagination is enough to make anything. And 3 germ layers can be given rise to. That is, the 3 germ layers is one example of a cell type derived from each layer. Ectoderm derived, mesoderm derived, endoderm derived. We have got 3 main germ layers, and it can give rise to each and every cell types that is present in our body. Differentiation of the embryonic stem cell. When removed from the factors that maintain them as stem cell, that is, the factors that help them to have the stemness is removed, it will differentiate, and under appropriate conditions, any insult is enough. Just take the petri dish, keep outside for some time. Slight increase in temperature happened or decrease in temperature happened. That is good enough. Any insult if you give to the embryonic stem cell, it will differentiate and generate a progeny consisting of derivatives that the 3 germ layers are consisting of. That is, the mesoderm, endoderm, and ectoderm derived cells can be formed from the stem cell if you stop producing these factors that allow, or the feeder layer factors that allow the stem cells to remain as stem cell. General approaches of differentiation. ES cells, that is embryonic stem cell, that is in short form it is called ES cells, are allowed to aggregate and form 3-dimensional colonies and known as embryoid bodies. Embryoid bodies are a formation is a unique technique that is used in some cell culture field, because which can be fused. Embryoid bodies can be fused into another developing embryo, and you can make something called chimeric mice or chimeric animal. So that's they are called EBs, embryoid bodies. ES cells are cultured directly on stromal cells, and differentiation takes place in contact with these cells. And it involves differentiating ES cells into a monolayer or extracellular matrix protein, because they need to have some attachment. Without that, they can't grow in cultured, suspended culture like bacteria. It cannot grow. And ESC, embryonic stem cell differentiation. So you can have different methods of differentiation is there. And initiation of differentiation is getting rid of feeder layer and also getting rid of the leukemia inhibitory factor, LIF, which is a protein factor that allow the stem cells to continue to be in stem cell. So feeder cells also does more or less the same function. So LIF and feeder layer, you remove it, they are now ready for differentiation. ES cells differentiate into embryoid bodies, they look like this spherical, and cells differentiate into stromal cells, another method, and cells differentiate into extracellular matrix protein, and all cells that eventually give rise to differentiated cells, which we can analyze it. And tissue stem cells or adult stem cell. We have seen it, every tissue have got stem cell, which is for running the maintenance of that tissue or organ. So we can see where all the tissue stem cells are there. Where do you find them? Surface of the eye, it has, because you have got cornea, conjunctiva, etc. Brain stem cells we have, skin stem cell we have, breast stem cells we have, testicles we have, because the germs you have to produce a stem cell. Intestine has got intestinal stem cell, bone marrow has got bone marrow stem cell, and muscles have got stem cells. So these are all running the steady state affair. They are not meant for just making a new set of muscle, but they are running the show, homeostasis of that organ. And adult stem cell, in short form, they are called ASCs, and they have certain characteristics. They have potential to self-renew for a long period of time. They can give rise to mature cell types that have characteristic morphologies and specialized function along multiple lineages. So they are quite unique in that way. They will continue to give rise to that cell type, what it is supposed to make it throughout the lifespan of that organism. Types of ASCs: hematopoietic stem cell, mesenchymal stem cell, and other stem cells such as neural stem cell, endothelial stem cell, intestinal stem cell, olfactory, adult stem cell, mammary stem cell. It's even your hair follicles have got stem cells, so that it can give rise to new hair strand. Sources: they are bone marrow, umbilical cord, cord blood, and adipose tissues. Lot of tissues, deciduous tooth, brain, peripheral blood, they all have good source of stem cells, and whether we can harness them and give rise to that tissue is limited by the availability of suitable technique. Adult stem cell, you can see like this. This is the bone. Bone has got bone marrow stem cell, and you can give rise to the hematopoietic stem cell and multipotent stem cell, and it give rise to myeloid lineage or lymphoid lineage. And lymphoid lineage, it give rise to bunch of cells, that is natural killer cell, T lymphocyte, B lymphocyte, etc. And the myeloid lineage give rise to neutrophil, basophil, eosinophil, and monocyte, macrophages, platelets, and RBCs. And on the other hand, if you have got a stromal stem cell of the bone marrow, it can give rise to lining cell and also osteocyte, and also it can give rise to a dedicated cell types, as you can see here, hematopoietic cell and marrow, adipocyte, etc. Because the bone marrow is not just for producing the blood alone. It has got adipose tissue, it has got calcium producing cells, etc. So blood stem cells, they are a classic example of multipotent cell, because they can give rise to a group of cell types. It undergoes differentiation, and you can see here, and only specialized types of blood cells, that is red blood cells, white blood cells, and platelets, are usually kept in circulation, but their original source is from the bone marrow stem cell. And types of adult stem cells and their hierarchies. So principles of renewing tissues. So what is the principle? How do we decide, oh, it is time to renew or it is time to make it? It is purely demand based. Say you donated blood to someone. They took 300 ml of blood from you. So this is basically nothing but an injury. Injury means, although there is no physical injury, there is a depletion. In every injury, say your liver got damaged, what is happening? Tissue loss. Your leg got hurt, so basically there is a tissue damage. So the removal of the blood from your body also is nothing but depletion of tissue. Immediately it will be sensed by your bone marrow cells, and they start overproducing. The rate of synthesis will be more. Same way in females who have got this menstrual cycle, they lose blood from their body, although it is not large quantity, but again immediately it will start producing more of the blood in their body just to balance out the lost blood during their menstrual cycle. So the stem cells have to self-renew, divide rarely, means basically based on demand, and high potency rate. High potency rate means they can divide efficiently and completely and with the adequate number. It cannot happen if there are 100 stem cells or only 30 are dividing and remaining 70 are just lethargic. They may or may not divide. It doesn't happen. They are highly potent. They are capable of dividing. Earlier example I told you 30 cells, because 30 only you need, because if only 30 cells are damaged, you don't need 100 stem cells to divide now. Only 30 cells will divide, make a copy of themselves, and then the remaining newly formed 30 will now give rise to the differentiated cells. And the committed progenitors, they are transiently amplifying cells. They are multipotent in nature and they divide rapidly, because the actual stem cell make a copy if needed. But once they made a copy, that is the committed progenitors are made, now they have to rapidly divide. Like when someone removed the blood by blood donation, you have a depletion of cells. Your bone marrow cells give rise to some of the committed cells, which is capable of giving rise to WBC and RBC. Once it is been given rise, they will divide fast. They won't say, okay, I will divide in next 10 days. No, within 2 days or 3 days it is done. The lost blood is restored back and they divide rapidly, and they do not have the ability to self-renew because they have to come from the stem cell. And committed progenitors are just meant for dividing as progenitor, and they continue to differentiate. They will not make a copy of themselves and keep it. If committed progenitors, 100 committed progenitors are formed, they will become 200. But of the 200, the entire 200 give rise to the differentiated cell type. They will not keep 100 copy like a stem cell is doing. So this is some difference which you should keep in mind. And then they give rise to the specialized cells. That is, they are meant for work, and many of them, they will not divide at all. And hematopoietic stem cell, if you see here, this is exactly what happens. They have got different lineage. Major lineages are lymphoid and myeloid lineage. You can see here in one group, they give rise to NK cell, T cell, B cell, and another group, it gives rise to dendritic cell, and then megakaryocyte, platelets, erythrocytes, macrophages, neutrophil, eosinophil, basophil, and these group are called committed progenitors. And bone marrow stem cells are the actual stem cells. As you can see here, this is the real stem cell, and these committed progenitors you are seeing here, these are committed progenitors, and they can expand in number. Usually if you have taken a vaccination and if that it is staying in the memory, then if you encounter that pathogen again, there will be a sudden surge of these committed progenitors that will kickstart, so that they can give rise to the specific cell type that is needed for producing the antibody against that pathogen, which is normally the B cell, which produces the antibody here. And as you can see here, these are specialized cells which are not going to divide. They are going to perform their duty. So committed progenitors can divide, and they won't make a copy of themselves. Of course, they will make a copy of themselves, but they will not store them. They are meant for differentiating. And then comes the mesenchymal stem cell, and this is also present in the bone marrow, and they have also a group of committed progenitors, and the specialized cell give rise to the bone. That is, bone marrow need the maintenance of the bone, and also it need to maintain the cartilage, that is called chondrocytes, and then the fat storing adipocytes. So the mesenchymal stem cells of the bone marrow, they are giving rise to mainly the bone, cartilage, and the fat producing cell, fat storing cells. So these are all the mesenchymal stem cells main job. Hematopoietic stem cells are the one which give rise to these bone marrow hematopoietic stem cells give rise to the blood related, which are meant both for your oxygen transport and also for the immunity. Then comes the neural stem cell, which is present in the brain, NSC in short form. And they also have got a phase of committed progenitor phase, and then they can give rise to the neuron, interneurons, oligodendrocytes, type 2 astrocytes, type 1 astrocytes. These all are specialized cells, and we know neurons do not divide. So oligodendrocytes, these are all glial. Of course, peripheral nervous system has got Schwann cells, but this we are talking about the brain stem cell. They are called the neural stem cells. Hence, oligodendrocytes are there. They are not neurons, they are glia. Whereas neurons and interneurons are usually found in the spinal cord, and the neurons are present both in the brain and spinal cord. And outside this brain and spinal cord, we usually refer to them as peripheral nervous system. They are not central nervous system. So oligodendrocytes are seen only in the central nervous system, but which is not neuron, but it's a glia. So what you should understand, these neural stem cells are capable of giving rise to both neurons and glia, which are terminally differentiated, and astrocytes also a glial cell population. And then you can see the gut stem cell. Its short form, it is called GSC. And gut stem cell, they are present mainly in the small intestine, because that is one organ which is having maximum wear and tear. Wind signaling comes into picture, wind signaling maintains it, and wind signaling is strictly controlled as well. Otherwise, you can have over proliferation. So that also, proliferation is needed, badly needed, but you don't want over proliferation. So intestinal stem cell is a very strictly controlled stem cell, as well as the proliferation group of cells. So here you have committed progenitors, and they gives rise to Paneth cell, because every cell present in the intestinal epithelium, the gut stem cell should be able to supply. Paneth cells, goblet cells, and endocrine cells, columnar cells, which are mainly the specialized cell. Each cell have got its own function. Like endocrine cells produces lots of hormone, and many intestinal cells produces serotonin, and goblet cells produce the mucus. They have dedicated biological function. And now comes induced pluripotent cell, something very exciting topic that has fetched a Nobel Prize. Yamanaka has got Nobel Prize for the discovery of it. That is very simple. The question was, can I make a stem cell on demand? I have a cell, I'm ready to share my skin or my buccal swab, and I'm ready to donate. Anyway, it doesn't cost me anything. Just swab your buccal cavity, you get it, your cell. Can you make that into stem cell? So this was a very interesting question. They have done extensive research. They narrowed down from 256 down to 4 factors. Just 4 factors is enough. And OCT4, SOX2, KLF, and MIC. So these are all the Yamanaka factors. So what you can see here is induced pluripotent stem cell. What they do is take a cell from the body, preferably fibroblast, which is easily available. Genetic reprogramming, that is making the cell to go back all the way back into embryonic stage, and add certain gene to that particular cell, and you end up getting induced pluripotent cell. Behaves like an embryonic stem cell, and that can be cultured just like use of feeder layer or leaf, etc. And that is being trained to differentiate into any cell type, neuron, muscle, or skin, anything. As you name it, you can give rise to. And induced pluripotent stem cells, they are basically IPCs, which are, as you can see here, they are cultured and cells from the body. You collect them, and then you do genetic reprogramming by transfection. Like I told you, OCT4, SOX2, KLF, and MIC. KLF4 usually, and MIC. These you express in the cell by transfection. Then it is going to behave like a stem cell. You end up getting stem cell, and that can differentiate into bone, skin, muscle. Only 3 are shown here, but don't think only 3 can be given. You can give rise to any type of cell type. So what you understand about the stem cell so far is stem cell has got the inherent capacity to differentiate into any cell type in your body. Say a person has got a tissue damaged, and now a scar is formed by fibrosis, but you want a proper tissue that is formed present in that organ. Stem cell have the capacity, provided the environment is supporting that. If the environment is not supporting the stem cell's ability to form, then it will fail, because stem cells cannot do the job of that particular tissue. Stem cells can give rise to that particular tissue. So the environment should be supportive of that. So we will study more about the stem cells and also about the regeneration biology in the next class. Thank you.